Okay, so you hit it once, bam, 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 all the ones, all the squares with one dot means that they are wet. Okay, and that represents a single hit, okay, single target cell. Yes? Okay. On the other hand, if you have another area of the park where you have tile, but the tile has different different colors. Let's suppose it has different colors, but it's the same tile, but the tile has been split in two, and one side has one color, one side has the other color. That represents the multi-target single hit. What it's saying is this, that if that square, one half gets hit, boom, is not considered to be wet. So, all of these squares that I have here, are they wet? No, they're not. They are not considered to be wet. They will only be considered to be wet when you hit the other side. Now they are wet. And that represents the multi-target. Okay? Why do they call it single hit? Is because you have to hit it one time to that area. Why? Because there are other, remember, all of these are models. This is not reality. I mean, it's possibly, yeah, for some organisms, but there are other molecules that might have that you need single target, multi-hit. Okay? You following me? And so these are just models, and the only two models we're going to look at are these two: the multi-target single hit and the single hit multi-target. So that's what this represents. This represents multi-target, single hit, and this represents single target. Okay? And now what you do is statistically you look at the numbers and then you develop graphs, and that's what we are seeing here. Okay. All right. So we have this extrapolation number is used in case you want to follow this line and you see how the survival, statistically how it will be, but in reality it's not like that. In reality, what you're looking at, you're seeing this. Okay? Now, this DQ is important because it's a threshold dose. Okay? What is it saying? It's related to the capacity of the cell to recover from some lethal damage, which means, okay, up to this point, if you hit it, up to this point, with this dose, you hit a colony of cells. With this dose, from here to here, nothing happened. Okay, you hit him once. You hit one side of the square. Okay? Once you start going beyond this side, now you're hitting it twice. And now the squares are believed to be wet. Now the squares, okay, which represents organisms, complicated organisms, now their survival goes down. Okay? Now, to find, just like it was related, this D0 is the necessary survival to 37% in the straight portion of the line, represents the same 37% that we looked at here on the single target, single hit. Okay? Do we need to explore that number? Not for us. A radiobiologist looking at how an organism behaves, yeah, it is important for them to look at this. But for us, no, it's just a concept to understand. Are we good? All right. Did this help?
Yeah.
Okay. All right, cell cycle assessment. Okay, in humans, cells replicate by mitosis. The average time from one mitosis to another is called the cell cycle time, or the generation time. Okay. So one mitosis to the next mitosis, that's going to be the cell cycle time, okay, or generation time.
I think.
Uh-huh.
Yeah, may I have a question? This is for
Yeah.
if the second is the multi target, but single hit, right?
Single hit.
Single hit, two spot.
It means means single hit because each spot, okay, each side in this example, each side of the square has to be hit once. Okay? Right? And like I told John, right? Why is it called multi-target single hit? I don't know, but I can try to explain it, and that is, there are organisms out there that they have multi-targets. And you need multi-hits. Okay, so what it's saying, when I say multi-target, multi-hit, what it's saying is, if we had this, then you will need to hit it a second time or third time. I don't know how many times. Okay, that's when it's multi-hit. Most organisms behave more like this, more complicated organisms. Humans, okay, multi-target, single hit. In the case of humans, okay, in the case of humans, once again, you just believe that number keeps changing so often. What it is you have okay. It'll be like, so you have multi-target. This is one big square. And for the cell to be annihilated, you will have to hit it 14 times, or 19 times, or 7 times, depending on the literature you look at. Okay, for it to die. That makes sense? This is part of this big square. This is one little square that I just magnified. Okay. All right, but it has multi-targets, and so each one of them will have to be hit for the cell to be annihilated. Annihilated means killed. Okay. That makes sense? Yes.
Are they repairable if
They survive. They survive. So how are they doing it? Either by repair or because they don't need that portion of the DNA? No.
thought the DNA gets rearranged if they get it multi-hit? And then if it's for single hit, that's when we lose like one arm of the chromosome, right?
Yeah, but in this case, take a look. In the case of humans, you have so many targets, okay, that you will have to hit each target at least one time. Okay. The other thing here is that if, say, you're hitting this target and you hit this target 3 times, because it's a random, right? It's a random event, isn't it?
Yeah.
I mean, we don't know where radiation is going. Let's say you hit this one 27 times, right? What happened to that target?
No.
You only need
This
one,
it mutates.
so it's
Yeah.
wasted. That's wasted radiation. Okay, you only need to hit it once. Okay, so, but each one of them, each one of them will have to be hit. Okay, each one of them will be hit one time, that's all they need. Okay, and then boom, the cell is gone. That point. If there is still one surviving, the cell can survive. 2 surviving, the cell will survive. You have, say you have on that area, but you still have those 2 targets empty. They're still dry. You haven't been hit, the cell will survive.
It survives, but it may be totally mutated, right?
No,
No?
the cell will survive. The cell will survive. Yeah. Yeah. And is it because, and once again, we go back to the DNA being so big, and lots of the DNA in humans, okay, it is believed, maybe 50 years from now will change, but it is believed that it's garbage DNA. DNA that is not coding for anything.
But what about all those situations where the DNA
Is
is
hit.
broken or breaks the chain? I mean, that's a mutation, right?
But it's,
But
but
we're
it's
just
not,
looking
but
at
it's
if it
not
lives or
coding,
dies.
but if it's not coding for anything, that's the problem. It's mutated, but it's not
But
coding for
it's just
anything.
as likely to hit important stuff as it is non-important, right?
What happens with human DNA is, you have this huge change, right? DNA. Think of this as the double strand. All right, this is just a tiny little portion of human DNA. Out of this portion, okay, only this codon is important. That's it. Only this one. So you can hit all of this. You can hit it. So what? Mutated, it's ruined. You make more of the ruined one. So what? It's not coding for anything. The problem is when you hit this one. Boom. Then is when you have a problem. Why is it like that? We don't know why is it like that. Scientists, they think it's because of the way humans have grown and evolved in this planet, that it was probably a protection against viruses. Because viruses can invade your DNA, okay? And they can come in and inject their bad DNA, okay? And if they inject it into one of these, no problem. The problem is when they start changing this. And not only viruses, there are many other agents that can change. Radiation is one of them. So it's a protection mechanism. Okay? It's a camouflage. Yes, Gary?
So you're basically saying for the multi-target hit is that a cell has to be hit 14 times in order for it to die? Is that right?
And it's not that the cell has to be hit, the targets.
The target
Targets.
DNA. Okay.
The targets, not the cell, the targets. The cell can be hit 100 times, but if you're not hitting the target, right?
That
You
important
can be hitting the
DNA.
cytoplasm, big deal. You can make more of it.
Okay, so what I'm trying to say.
The point here is the target. If you're hitting DNA, then it's the problem. That is the problem.
So DNA has to be.
Go ahead.
So DNA has to be hit 14 times for the cell to die. If they, okay.
In the case of inhumans. Okay, but remember what I said, that number is not set in stone. I've seen it change going all the way up to 19, I've seen it going down to 7, 8, 9. Okay. But most people believe it's about 14. Yes?
Oh, well, by DNA, you mean the codon, actually, that's the critical one. The DNA itself, right?
I don't know. I mean, the target molecule is DNA. The key molecule is DNA. That's what it's called. All right? So I don't want to call the specific codons because are we okay? Good? Yeah? All right. Okay, cells in mitosis are always the most radiosensitive. Why do you think is that true?
It's when DNA is replicating.
Not in mitosis. By the time you get to mitosis, replication has already happened.
Oh. When DNA is divided, when DNA is being divided into daughter cells.
Question? Angela King, what do you think? Why do you think cells are more radiosensitive? And what is radiosensitive? Are we clear with that?
More sensitive to radiation.
They're more vulnerable.
All right, there you go. Thank you. Yeah. All right. Why are they more vulnerable during mitosis?
I thought it was because that's when they're dividing and they're usually younger cells.
Good idea.
They're not mature. So, I don't know.
John?
Is it because the DNA is all condensed in the chromosome?
Exactly. That's because by then DNA, which was all spread out, remember, before mitosis, during synthesis, it's all spread out. Now, during mitosis, it has condensed. And now, condensed, how do we call condensed DNA?
Chromosome.
Chromosomes. So now you have this on a cell. All right, when you look at a cell, right, and during synthesis, let's look at the nucleus. During synthesis, you have DNA everywhere. And it's all spread out. And it doesn't mean that the cell is not sensitive. It is sensitive, okay? But when it gets to mitosis, when it's just about to enter mitosis, here, okay, here it is. Now, all of this stretch, remember what I had called miles of DNA? Now, those miles of DNA are gonna be packed. They're gonna be, there, you pull them, okay, and now all of this is going to be set into chromosomes. Another chromosome, another chromosome, another chromosome. So all of that DNA now is concentrated. Everything that was all stretched now is all coiled up in these chromosomes. So now you have lots of DNA in one location, in a very small area. Radiation comes and hits it, it's gonna do a lot of damage. Okay, it's gonna do a lot of damage because it's hitting multiple areas of DNA. That make sense? Okay, good. Alright, so this is what we find. When we look at cell surviving, or cell survival, what we find is this, okay? This is the surviving fraction, okay? And these are the stages of, this is from Bouchon, by the way. These are the stages of, different stages the cell is going through. This is mitosis. This is growth 1. This is synthesis. This is growth 2. Then mitosis. Okay, this is the continuation from here to here. So this is one continuous thing. So from here, continues here. And so, I mean, take a look. The survival fraction, as you can see, is more sensitive, okay, during mitosis. See that? During mitosis. Okay, see these numbers here? That means that less and less cells are surviving during this time. And that's because why? All the DNA is condensed, okay, in one small area, in the area of the chromosomes. You hit a chromosome, you're hitting not one base or two bases, you're hitting chunks of DNA. Okay, make sense? Mm-hmm. All right. All right, let's go back to LTE, RBE, and OER. So, to start with, Dennis Martinez, tell me what is LET?
The linear efficiency.
Nope.
No.
WEU. What is LET? And what does it represent?
I think that his answer is wrong.
No, it's not. All right. Ms. Morales, what is LET? And what does it represent? No?
Like ionizing radiation? Like a measurement of ionizing radiation?
What is it measuring?
Ionizing radiation.
All right, Jesus?
High LETs,
But what does LET stands for?
linear effect threshold.
Oh. Good guess. All right. I know you look at it. What is it? LET. LET. Okay, read us what you read. Yes. Monique. So linear energy transfer. Linear energy transfer. Okay? Not linear effectiveness, nothing like that. No. Linear energy transfer. All right? And what does it represent? What does it represent? What is it telling us?
The dose that's absorbed, that goes through the tissue.
In what?
In a human. or
Okay,
animal.
but it's the track.
Yeah,
Okay.
unit track.
The unit track, okay, and how much dose was deposited, how much energy is deposited in a length, in a specific length. a millimeter, a micrometer, a centimeter, what was it? Okay, that's what LET is. All right, what is RBE?
Relative biologic effectiveness.
Then what is that?
The capabilities of radiation with different LETs to produce like a reaction.
Good.
Okay.
Okay, we haven't done it yet, but that's good. All right. Okay. Ms. Valentina. Okay, what is over here? What?
Is the oxygen effect?
Oxygen effect, okay. And what does the E stand for?
Efficiency?
No.
Enhancement?
Enhancement. And the R? Ratio. And what is that saying? Rachel Kreider, what is it saying?
The ratio of radiation dose required to cause particular biologic effects.
But in your own terms, with that one, that sounds like me.
Yeah.
You even had my accent there. More like you. Yeah. What is OER saying? What is it telling us? But what do you remember? Well, listen to the definition of what it's saying. Oxygen enhancement ratio. What did that say?
To me, it would be like if the oxygen was increasing the ratio, it would be oxygen.
There you go. That's it. That sounds like you. Exactly. Good. So what it's saying, if oxygen is present, if oxygen is there, okay, during an ionization event, it will be enhanced. Okay, that's what it's saying. Are we good?
Yes.
All right, good. All right, so let's take a look here. Okay, so take a look. What we observe, and let me see, I have another one here. Let's start with this one here. Look what happens when you have low LET and you have high LET. All right, look at this. Look what happens when low LET is present, and this is radiation dose. Low LET. If oxygen is present, okay, look at this. You enhance the radiation, which means that less dose will do more damage. That's what it's saying. Listen carefully. Less dose will do more damage if oxygen is present. Okay, see that? If oxygen is present, you don't need, 1,800, I don't know what is it, whatever radiation units they are using. Let's say 1,800 rats. You don't need that to create the damage that you can do with only 1,000 when oxygen is present.
Okay.
Make sense?
Yeah, so if it's like if oxygen is present, the radiation damage to a cell is greater. Is that what I'm saying? Okay.
Mm-hmm.
So in human cells, is oxygen always present, or does it depend on the cell? I mean, say in a bone, is there less oxygen, so it's not as effective, or?
Yeah, and those that are using the most oxygen, they're going to be more radiosensitive. And here we can think of this because they use that concept when they are dealing with cancer cells. Okay, because cancer cells, you will think, since they take so much blood because of the nutrients, you will think that they are in a very aerobic environment, and they are. But the problem with tumors is the following: you have tumors like that, and you have cells like that everywhere, right? And so if you are dealing with a tumor, yeah, the surface of the tumor, yeah, it does take a lot, but the inner part is very anaerobic, and that's why it's so difficult to kill them. That's why it's so difficult to kill cancer cells. You kill the surface, but then the inside is still alive. That's just the best example I have when when oxygen is present and when it's not. So let's go back here. Now, take a look with high LET. With high LET, depending on the energy of it, oxygen doesn't really do anything. Doesn't really do much, especially when you have already killed the cell, right? So oxygen doesn't do much, but it does a lot when oxygen is present and you're using low LET. Okay, remember earlier we had talked about with low LET, the oxygen enhancement ratio is really important. Yeah, okay. With high LET, it doesn't really matter because it has already done much damage. Let's see what in here. That's fine. Okay. All right, we're done. Okay, this is the end of our section. Okay. All right, so remember, when is our exam?
Monday.